and living things.
Hey, look at that. It looks like bear.
All right,
Where are we writing this?
you don't have to write anything down. All these slides are basically right from your notes in your note packet on scientific method. But I just want a chance to talk about it again, summarize, review, and then if there's anything you have a question about, you could ask a question. All right, so scientific method. So when we talked about the scientific method, we said there's sort of 6 steps that we named that are important. Problem, research, hypothesis, experiment, observation, and conclusion. People really hate elephants on cars. And so this is basically the steps the scientists follow when they want to learn some new information about something. It starts with the problem step. In scientific method, the problem step is when you ask a question. Okay? Scientists pose a question that they want answered. That's the first step. After they have a question that they're looking to answer, second, they do some research, some background information. Find some information about that topic that they're going to be studying so that they can make an intelligent hypothesis and set up an experiment properly. I put your homework away and other stuff. I want you to just pay attention and care. Put that away. So, we gather some basic information, and then it's time for the hypothesis. Now, I know you guys always want to say hypothesis-educated guess, but again, what it really is, it's when scientists know what they're going to be testing, and their hypothesis is what they think is going to happen. Their prediction about what's going to be the result of their experiment. You always predict what's going to happen before you actually do the experiment, and that's the hypothesis. Once you have a hypothesis, then scientists can set up an experiment that's going to test it. And we talked a lot about how to set up a good experiment. They set up different groups, and certain groups get treated differently. And you collect some information, and you use that information to answer your question. So in the experiment, you basically test your hypothesis. You say, all right, so I predict this is going to happen, and you set up an experiment that's going to tell you if that's true or not. After the experiment is set up, you are recording some data, gathering some information. That's the observation step. Observations are the data that you gather. And finally, when you have these observations, you can come up with a conclusion. And in scientific method, the conclusion is, was your hypothesis correct or was it incorrect? You decide, based on the data, if you were right or wrong in your prediction. And so what we did, and what you're going to have to do on the final exam as well, is know how the scientific method works and be able also to design an experiment or read about an experiment and know how to set up a proper scientific experiment. Another word that's important, inference. So observations are things you actually see or sense or measure. An inference is a little bit different. An inference is some sort of conclusion that you make based on those things that you see, based on that information. When we do set up our experiment, okay, again, we have different groups that we set up, and we have these variables. You know, the independent variable, variables held constant, and the dependent variable. So the independent variable in the experiment, a variable is just sort of a factor in your experiment. You can measure something, you can change. So the independent variable, this is the one thing that you're actually testing. If you remember, the I looks like a 1. It's the one thing you want to be different in your groups that you set up. Okay? And like we said, a good experiment has only one thing different. You can think about it as sort of the cause in the experiment. Good. And then we have variables held constant. So if we only want one thing to be different between our groups, that means there must be a whole bunch of things we want to be the same. And those things are all the variables held constant. And the last one is the dependent variable. This is the data you collect, the information you gather that's going to help you to make your hypothesis. You don't know it ahead of time. Sort of you collect this data as the experiment's going along. It's the results of your experiment. Any questions about those variables? So I looks like a 1 because it's the one thing different. D for data. All right, so when we have our experiment, we always have to have at least 2 groups. You always have one control group at least and one experimental group. Sometimes there's more experimental groups, but at least one. The experimental group is a group that you change something. You alter the independent variable. You give the plants extra fertilizer, or you feed the mice vitamin C, or you keep a plant in the dark. So you're changing something about its normal conditions. That's the experimental group. The control group is sort of a test run, a group that's sort of just treated normally. You don't change the independent variable, and you always need that control group as a comparison. You're always going to compare how the experimental group did versus the control group. You need that baseline to see if there was any actual effect. Okay, any questions about scientific method, experiments? Okay, second thing we'll review today, living things. So living things have certain characteristics that all living things have. All living things are made of cells. Sometimes it's a single cell, like a bacteria. Other times it could be trillions of cells, like a person. But all living things are made of cells. They all need to obtain and use energy in some way. Living things grow and develop. They respond to stimuli in their environment, they reproduce, and they are all adapted to their surroundings. Those are the characteristics of life. And all living things need certain things in order to stay alive. They need a source of energy, they need water, they need something from the air, space to live, and proper temperature. Now, every year when we look at the results of exams and tests, this is one area that students almost always have a difficult time with. It's one of the most difficult topics and most commonly missed topics on the test. Photosynthesis and respiration. I think you guys understand pretty well, but you need to know, not going to have to necessarily write out the formulas, but you need to know about both of these processes. So let's start with photosynthesis. It needs 3 things. The process of photosynthesis happens in cells. What's one thing, Grant?
Sunlight.
It needs light. What else, Sam?
Water.
Water. And one other thing, Garrett?
Carbon dioxide.
Yeah. It's light, water, and carbon dioxide. 3 things are the reactants. And if those 3 things are there, then a cell can produce 2 things from those. What are the 2 things, Alexis? Glucose. And what's the other, Nina? Oxygen. So that's the process of photosynthesis. Mainly it's plants that do it, but some other things as well. And it happens in what organelle?
Chloroplast.
Chloroplast.
Yeah.
Happens in the chloroplast, green organelles in plant cells. All right, so this is, when we say plants make their own food, this is what they do. They make glucose using energy from the sun. Respiration is another process. It doesn't just happen in plants, it happens in basically all living things. Okay, what is needed? What are the reactants in respiration, Kevin?
Oxygen.
It needs oxygen and glucose. And if those 2 things are present, then this process can happen. And it produces 3 things. Samantha? ATP, that's what? What is ATP? Joe?
Automatic toilet paper.
What is ATP?
Energy.
Energy. It's the type of energy a cell can actually use. It also produces water, carbon dioxide as waste products. What organelle does that happen in?
I got this. Kevin thinks he's got it, no,
Sydney? Mitochondria happens in all cells. Mitochondria. And again, plants make glucose using photosynthesis, but they still need to go through respiration so they can take that glucose and turn it into ATP. Any questions about those? Organelles of the cell, you should be able to identify a few of them. There's a star by the most important ones.
You got this one.
And these are the easy ones, really.
I got this one for you.
So, nucleus, chloroplast, cell wall,
What's the Golgi apparatus?
Golgi bodies. Yes, I mean, no, I mean, it's almost impossible to tell a lysosome from a vacuole. They're just, so no, you don't need to know lysosome.
We have to know Golgi bodies.
Not, I don't think there's a whole lot, no.
Okay.
These are the big ones. Okay, nucleus, obviously it's one of the largest organelles, generally center of the cell. Chloroplast, only in plant cells, they're generally around the outside. They're large organelles, they have discs inside of them. If it was in color, they'd be green, right, so?
How do you know the difference between endoplasmic reticulum and ribosome?
Well, the ER are the folds, the membrane. The ribosomes are actually dots on it. So ribosomes are small little circles. What does it do? Photosynthesis.
What's this section?
Don't worry about it. Helps the cell divide. All right, and then last thing, evolution and natural selection were part of this unit. So remember, evolution is a change in a species over a period of time, and it happens through this process of natural selection. In natural selection, we know that not all members of a species are exactly the same. There's some variations, some differences. And if one of those variations happens to be beneficial for that individual, they can do what? Have a beneficial trait. They can survive, they can live. And if they can live, that also means they're most likely able to reproduce. When they do reproduce, they pass on those traits that help them survive to their offspring. And over a period of time, generation after generation, those variations become more and more common, and over time the species all have it. And that's how species change over time, through this process of natural selection. We call those traits they have adaptations. So adaptations that allow them to survive.
I see you have your Burn Dairy Couch. You get your free coffee today.
I did not.
You get free coffee?
I made coffee at home.
On Tuesdays.
A couple other factors here. These two things are important. How quickly a species can change depends on two things, really. So it depends on how quickly that species reproduces. Some species reproduce very quickly. If you think about like a bacteria, fruit flies, a protist, they can reproduce in a matter of days, or even sometimes hours. So they can go through generations very, very quickly. Versus other species which take years to reproduce. Which one do you think would be able to adapt more quickly?
The quicker.
Bacteria, protists. The things that reproduce more quickly can adapt at a faster rate because they go through many generations very quickly. Also, if the environment is changing, that leads to more rapid evolution as well, because changes in the environment may make different variations more beneficial. Also, important thing is that if a species is unable to adapt to a changing environment, that may become extinct. Yeah? I don't know if it's necessarily on there, but who is it?
Charles.